Okay. Why did the detective stay in bed? Why did the detective stay in bed? Any ideas? It was a long Super Bowl. You guys are rusty, huh? He wanted to stay undercover. Why are parallel lines so lonely? Because they never intersect? Wow, that's a big word. Is that what you guys say? Let's intersect sometime. They never meet. They never meet. Intersect is accurate, but that's kind of awkward. All right. What did one elevator say to the other? What did one elevator say to the other? Going up or down? Okay. What's up? I think I'm coming down with something. Okay. All right. All right, let's look at the syllabus real quick. So we are going to be in tissue repair, fibrosis, and healing this week. You've got post-quiz number 3 due on Friday. And then next week we're in the immune system, and then we have our exam. Okay? I know. It's a lot of material. So here's what's going on. If we've had enough material thus far where you should have a pretty firm handle on it. If there are big gaps, let me give you an example. Like, I don't understand how inflammation is related to wound healing. You keep saying that, but I don't get it. That's kind of a big gap. You need to get that answered. So that's an opportunity to go to SI, ask that question. You can come into my office hours. We can go over it. The point is, don't wait until President's Day, which is like 2 days before the exam, where now you've got like 5 pages of I don't understands, when you can start getting the answers to the I don't understands right now. That's kind of the whole point of the pre and the post quizzes. It's forcing you guys to pay attention to what you're not getting and what you are understanding. You guys with me? So, when you cram right before an exam, like 2 days, there's so much material, it's literally, there's not enough time in the day. And it's not the best formula for success. There's lots of studies that talk about that. And I know you guys might be thinking, well, I'm a crammer, I've always done it that way. So yes, I'm trying to encourage you to change your habits. And that's why the class is designed the way it is. So you're already slowly, don't even realize it, but we're training you on how to study a little bit more consistently over time. So now's a good opportunity to take inventory and say, okay, what am I not understanding and what do I need to nail down before President's Day? Okay? And then I think what you'll see is your performance on exam 1 will be far better. Fair? Okay. So exam 1 is that Wednesday. We don't have any other assignments that week. And then if you look at the second section of the syllabus, we've got exam 2 that happens to fall on a Monday, and then the Wednesday after exam 2, we don't have class. And give you that week off. So you just focus on the exam, okay? And then it'll be the same thing for the final. You might have other finals that week, I understand, because it's finals week, but trying to make sure that we're giving you kind of enough space in the schedule to do well on the exams. Questions before we get going? Okay. Tissue repair. Tissue repair focuses heavily on the process of wound healing or the mechanisms of wound healing. And I've talked about this. I've actually drawn a figure like this on the board last week. Okay, this comes out from an older textbook. It's a 1996 textbook, but it's kind of like the classic textbook in wound healing. And this is one of my more passionate lectures because this is a lot of what my research is on is wound healing. And so you're going to see a little bit today, but maybe more on Wednesday, a lot of the type of research that we do in the space of wound healing and wound repair. It's not just skin wound healing, although that's where we're going to start because it's, I think, the easiest introduction to wound healing. But wound healing happens in every tissue, right? Inflammation triggers wound healing and initiates it to start, and that happens in every tissue. But it seems for students that skin wound healing or cutaneous wound healing is kind of really the easiest concept to put your head around because we're all extremely familiar with it. You don't have to be old and decrepit to wrestle with a wound healing example. You've all had a wound, okay? Everybody's fallen off a bike, okay? Everybody's banged their head, everybody's twisted an ankle, fill in the blank. We've all experienced wounds, even in our youth. And so we don't have to be old and have all these comorbidities to really understand what does it feel like to have, something wrong with your cardiac tissue, or what does it feel like to have an aortic valve problem, or kidney stones or gallstones. And some of you do have some of those things, and you can relate to them. But all of us can really look at wound healing and say, in the skin, I get it, and I can see these different steps and stages as things move forward. So the early phase of wound healing and the late phase in part A is inflammation. So this is early inflammation and late inflammation. Not sure why it's doing that. You know what? Hang on one second. We ran into this in the beginning of the semester, and I think what it likes to do is search for my drive all day long. So we'll just get rid of it. Bear with me. All right, we're just doing that. Okay. I don't know why it does that. Anyways, so the early and the late phase of wound healing involves what cell types first? Neutrophils, very well done. That's the first responder. And what cell types are second? Macrophages, tissue macrophages. Where do the tissue macrophages come from? Where do they come from? They just spontaneously show up? Some of them are resident, some of them are in the tissue, but those are resident macrophages. How about the recruited macrophages that become activated? Where do they come from? The bloodstream. Very good. What cell types did they used to be? Leukocytes, specifically monocytes, circulating monocytes. How did they get out? Yeah, like that whole 7-step thing. Yeah, roll, tumbling, backflip, yes. Handspring, yes. Right? Margination, right? Rolling, tethering, adhesion, diapedesis or transmigration, right? So if that's, here's an example. If that is foreign to you, you need to look back at that stuff, okay? So that's how these cells get there. Why do we want those cells to come in? Why are they helpful? How does it enable wound healing? I agree, that's a totally correct answer. What's that? Cleans up the debris. Very good. What else does it do? What else do these cells do? They clean up the debris because they're phagocytic. They're phagocytes. What else do they do? They kill bacteria. If there's not always is there. You roll your ankle, there's no bacteria, so they don't need to do anything there. Okay, but they do need to clean up damaged tissue because you roll your ankle, there's traumatized tissue. Okay, very good. What else can they do? Can they blow a whistle and signal other cells to go or do something? Yes. What kind of things do they trigger to happen, these macrophages? Angiogenesis. Very good. What is that? That's a big word. Creation of capillaries or new blood vessel elements. Why do you want to bring in new blood vessels? To bring in nutrition and like oxygen supply. You have damaged tissue, you need to repair it. That takes a lot of energy. So if you have a lot of energy demands, you bring in oxygen, which is your final electron acceptor, and if you run through aerobic respiration, you generate a lot of what? ATP, and that helps with the energy to fuel the repair process. Is this all making sense? Okay. What else can they do? Angiogenesis, very good. Can they signal any other cells to come in? These guys that come up, they peak, and then they do their job and they go away, if everything goes well. What other cells can you signal? Fibroblasts, very good. And what would you want fibroblasts to do? Let's say it's a skin example. What do you want those fibroblasts to do? Who said collagen? Make collagen, lay down new collagen. That's going to create maybe some elements of fibrosis or scarring, but you don't want this giant pit or hole in the skin. Okay? Is this making sense? So those macrophages are really important. And this is why there's this connection between repair and inflammation. It's important to have it to trigger all these things to take place. Okay? So if I lost you in that whole little speech, we need to talk. We need to talk. Richard, you should spend some time talking with Richard. He understands this really well. If you didn't really, a few of you that are answering the questions, great job. Others are quiet. I know you might be shy, and that's okay too. But if you're puzzled by what we just talked about, I expect, and by exam time, you should be able to connect those dots and integrate that information. Okay? Yeah, so the growth factors are manufactured by these macrophages. Some of them are angiogenic. Some of them trigger fibroblasts to come in and proliferate and lay down new collagen. The general term for that is a growth factor or a cytokine. They're chemical signals. And so you don't get those signals if you don't recruit the macrophages. Okay? So then here in part B, we've got what we call granulation tissue. Now, granulation tissue is called that because it looks granular histologically, and I'll show you some examples. It looks granular histologically because there's a tremendous amount of new capillaries that have formed. The inflammatory macrophages come in, they make all these angiogenic growth factors that produces granulation tissue that's populated with an extensive blood supply. And then this needs to go away, and then you have this phase of wound contraction and collagen accumulation. What cells make the collagen? We just said it, the fibroblasts. Now, there are skin fibroblasts. There are fibroblasts in most every tissue. So if you learn about what happens in the skin, you can kind of apply that wound healing step to every other tissue in the body. And that's why we use this as an example. Make sense? So black and white picture from the 90s. We did publish in color back then, but it was, I guess, less expensive to publish this one in black and white. But this hasn't changed. This hasn't changed. And modern day publications are referencing still this sort of pivotal book chapter. Yeah, question in the back? No? You forgot? Okay. If you remember it, raise your hand. Okay. I want you guys, hopefully you've spent a little time doing the readings. You did for the pre-quiz. I want you guys to pair up. I want one person to explain the difference between resolution and repair, and then the other person to explain the difference between first and second intention, healing by first or second intention. Ready? Go. Do you remember?
Is it just formed because like the fibroblasts make the collagen and then just collagen that's on the granulation? Is that what, like how else is it formed? Is it just collagen or what else is it?
Your question is about granulation tissue?
Yeah.
So how does it form?
Yeah. Is it just collagen?
No, it's tissue that's dense with macrophages, and they make all of these growth factors that trigger new blood vessels to grow, and so it has a huge population of new vasculature. I'll show you a picture. It'll be a little bit more obvious.
Okay, so it's a whole bunch of blood then?
Blood vessels. Blood vessels.
So
Mostly
it's not collagen.
capillaries.
So it's not collagen at all then?
Collagen comes later.
Okay,
Yeah.
okay, that makes more sense.
Yeah.
Thank you.
Yep.
So, I might be mixing these up. Resolution and repair, they're the same as like healing and resolution?
Right.
and
Yeah.
the resolution
Yeah.
is not going to leave it.
Yeah, you got it.
Okay,
Yep.
cool.
Yep. Yep. No, you're good. Don't be worried. All right, let's hear it. Some brave souls. Yes.
For person A, resolution is where it'll heal and not leave any mark. There won't be any tissue damage. Repair is going to leave fibrosis. So there's going to be scarring.
Yeah. Everybody agree with that? That is correct. That's a very solid explanation. So when you have the term resolution, think of regeneration, restoration of function. The function of the tissue comes back, wherever you are. So if you're in skin, the skin is going to close over, so it forms a barrier, so water doesn't leave. It creates a barrier so bacteria can't get in. There's sweat glands that are present in skin tissue. So resolution or regeneration is you get new sweat glands. Hair follicles, you get a restoration of hair regrowth, okay? Repair has scar or fibrosis. You don't have the return to function. You spackle the wall where there was a hole. And you can tell where it is because it looks different than the native drywall. Okay, very good. How about healing by first and second intention? Right up front. Yep.
Well, from my understanding, the first intention occurs when it's more like a clean cut, and the skin's connected to it. It's just connected, or I guess physically when you just close and just heal. And the second intention is when like bones like open to heal, like granulation and stuff like that.
Yeah. Very good. Did you guys all hear that? Okay, so first intention is usually a very clean wound, like a surgical incision. Okay, so you're going to have wound edges that are relatively easy to approximate. Well, that's a big word. What does approximate mean? Well, that means bring together. So a wound by first intention would be one where you want to bring these two edges together, and it's nicely going to approximate. A wound by second intention is one where you might have more of a hole or an ulcer. And so you really can't bring this over because there's not enough skin to stretch. Does that make sense? So you have to kind of leave it open, and it's going to heal by what we call second intention, meaning you're going to get macrophage infiltration, neutrophils, macrophage infiltration, granulation tissue, like she characterized, and then you'll recruit fibroblasts and it'll lay down new matrix. Well, you'll get all of that here, but you don't have to do as much of it. So scarring is more aggressive in second intention than first intention. Does that make sense? Somewhat intuitive. All right, well done. I can tell you guys are looking at the material before we show up. So let's talk on this slide where we look at, this says normal homeostasis up top. It looks better on the side screens over here. The best one over here, you can actually read it. Where you've got an injury to a cell and you've got a situation where you can either completely regenerate, and tissues that regenerate very well, you can see epidermis. That's why we're talking about skin. The GI tract. The hematopoietic system, which is our blood system. These are tissues that regenerate quite well. You could add to that bone. Bone regenerates very, very well. Okay, so I'd add bone to the list. In the situation of regeneration, like what we talked about, this is where you see complete restoration of function. So we have wound resolution. Over here, you can see there's a bigger gap right here. You get scarring, or in some chronic inflammatory diseases, you might have excessive fibrosis. Repair or healing, or what we classically refer to as wound healing, you don't have a complete restoration of function. Now, I would offer what you'll see clinically and what you'll see in your careers is that it's never one or the other. It's oftentimes some blend of the two. And as we get older and we have more comorbidities, you have more repairing that happens. And unfortunately, in some cases, you have more chronic inflammation, which leads to more or less favorable aesthetic outcomes. And when we're younger, you have much greater opportunity for complete regeneration of tissues. Okay? So, we talked about the fibroblast. Here's an example test question of sorts. What's the role of the fibroblast during the reconstructive phase of wound healing by second intention? Okay, by second intention. So you know we're talking about a pretty aggressive wound. You could expect there's going to be more fibrosis. There's more tissue that has to be relayed down. Okay? So what does it do? Does it generate new capillaries from vascular endothelial cells? Does it establish connections between the neighboring cells and help to contract their fibers? Does it synthesize and secrete collagen in the connective tissue proteins? Does it provide enzymes that debride the wound of dead cells? This is a clinical term, debride, meaning you remove, you scrape away, or you wash away, or you digest away the necrotic tissue. Or is it two of the above? Okay. Any takers? Max? Two of the above, okay. They do? Agreed? So that's correct. These fibroblasts play the role of both B and C, whereas they're laying down new collagen, but they're also facilitating or helping to close up that wound margin. And you'll get wound contraction that will take place. Okay? All right. So this slide, the importance of this slide is to talk about the role that cells and matrix play in a tissue. So all tissues have some combination of matrix and then cells. And the matrix, the analogy that I like to use is the matrix is like the jungle gym. And the cells are like the kids that play on the jungle gym. In the old days, they had like these dome structures that were metal, like a jungle gym that looked like a dome. And the kids would, you don't see them anymore because probably there's too many lawsuits or something bad happened. But the kids would crawl on this jungle gym, right? And that's really, I think, a good picture of what cells need, is they need an architecture by which to attach to. They just don't float in three-dimensional space. They actually attach to a substrate, or they connect to some sort of matrix. And typically, there's a matrix that's made up of collagen at some level. And there's other extracellular matrix proteins other than collagen. It's just one of the most abundant. Okay? So this is liver tissue, and we're blowing up in higher magnification this portal triad, so you can kind of appreciate the hepatic artery, the portal vein, and the bile duct. And that's this area that's right here. If you have injury to tissue and just the cells are compromised, but the matrix is left intact, if you go to heal, you're more likely to result in a regeneration or a restoration of normal function when the matrix architecture is left intact and you've just actually removed the cells only, or the cells have died. So what's the situation there? Well, if you have trauma, like a laceration to the liver, the chances of the matrix being maintained are slim. And so you don't necessarily preserve the architecture of the matrix. So when you bring new cells in, you have more excessive fibrosis. And this is true in most tissues. So all else being equal, if you've got damage to matrix and cells, you're going to end up with more fibrosis or scarring. If you have damage to just the cells but the matrix is left intact, it's more likely you can repopulate with new cells and get more of a restoration of function. So the important takeaway here is the matrix is very critical. The architecture is very critical. And when the cells are compromised, if their matrix environment that they lived on is left intact, you can repopulate that matrix and you get less scarring. Max?
Yeah. So does the matrix, when you get a new, like, influx of cells, does the matrix have some sort of, I don't know, I know this sounds dumb, but subconscious effect on the cells that kind of dictates their formation? Because obviously it doesn't have fibrosis, like they don't have that matrix, so they don't really have anything to kind of pull off of.
Yeah, so about 5 slides down the road, I'll address your question. Okay? It's a great question because these cells on this matrix are very complex. And I have a picture that might kind of like blow your mind a little bit. And if you took cell biology, you've seen this, but we'll get there. Leading up to that conversation, cell biology, is let's talk about some of the cellular cycles. So you all know, probably from high school biology, if you can recall, that there are different stages in the cell cycle. Sometimes the cell is at rest. Sometimes it's signaled to actually divide, okay, and proliferate. And then other times it might be signaled to migrate. Now, let's pretend that this is a fibroblast in the case of a wound, like in the skin. So we can enter, G1, and from G1 we get this duplication that takes place, and then there's a pause in the S phase. And the pause in the G1 to S phase, S is for synthesis, stands for synthesis, is to evaluate the integrity of the DNA and make sure that there aren't any mutations. And if there aren't any mutations, then it moves on to duplicate, and then you move into gap 2, and then you have a second checkpoint before you go through mitosis, before you're actually going to do a division. You've made another copy, and now you're checking to make sure that the copy had high fidelity. There aren't any mistakes. Then you move into mitosis, and you divide the cell, and you go from 1 cell to 2 cells. So if this is in a wound, and this is a fibroblast that's been recruited, this is happening again and again and again until that wound closes. And this is what's responsible for wound closure, is these fibroblasts go through the cell cycle, and it's like you're turning the crank again and again and again. So learning the events of the cell cycle are relevant to this class because this is how you close a wound. Now, I'm going to interject here. What happens if there's errors? What happens if there's errors? The healing process stops. What else? What happens if there's errors? Could lead to cancer. So we're going to talk about, we have a cancer section later in the semester, and we're going to talk about a little bit more detail on evaluating the errors or when problems arise. What could cause mutations or errors to occur? Exposure to UV radiation like sunlight, chemical, toxins, pollutants, okay? Basically our environment, the aging process, right? It's a disease, remember? So just the aging process, this cell cycle, I should go in the correct direction, this cell cycle doesn't go as well the older we get. In fact, it's been well documented that wounds take twice as long to heal in patients that are 70 years or older compared to patients that are pediatric up to about their middle 20s. So geriatric patients compared to pediatric patients, wound closure is twice as long because this thing isn't working quite as well. Okay. How do the cells signal to each other? So now we know principles about the cell. Now let's talk about how they communicate, because they all need to be in communication and doing sort of the same thing to work towards the same goal. So totally different biology, but anybody fascinated by like oceanography or marine biology? Like Planet Earth videos, or like you're skipping through the channels and it's like Shark Week and you just can't stop watching, right? Those schools of fish that swim together? Or maybe you're more into like avian biology. Birds will flock like this, and there are species of fish or birds that it's like as if we just decided as a classroom we're all going to do the same dance right now, and then we're going to go boogie out into the quad out front, and you can watch these fish move so fast in the ocean, to kind of distract their predators because they think that it's a larger organism. Some of them look like cyclones in the water. Birds will do the same thing. So in our body, our cells at some level behave kind of like a flock of birds or like a school of fish that are all kind of doing similar things. They actually have to be communicating to each other. And so they use different mechanisms of communication, our cells. They use these chemicals, these growth factors, these cytokines. If they release a signal and it binds to the same cell, it's called an autocrine response, meaning auto meaning self response. Okay? We'll see autocrine signaling being really important next week when we talk about the immune system. Because now we have these soldier cells that defend us, and we've got to have a way to activate them. Well, we'll see that autocrine signaling is important in that. Paracrine signaling, this one's probably a little bit more obvious of why it's important, is you need a neighboring cell to know what you're about to do, or what you want the neighboring cell to do. So if you release a growth factor or cytokine, it goes to a neighboring cell, and it causes activity on that neighboring cell by binding a receptor on that adjacent or neighboring cell. We call that paracrine signaling. And then last but not least, and we'll talk about this more in like our endocrine disorder section. Endocrine signaling is the cells release some signaling chemical. It goes into the bloodstream. It is dispersed distally or systemically and binds to distant target tissue, and this is called endocrine signaling. So this is the type of signaling that happens with sympathetic release when you're getting ready, when you get spooked, and all of a sudden your heart rate goes up. Okay, your respirations increase. This is what happens during endocrine signaling in mature females about every 28 days in order to get the endometrial lining ready for fertilization. This is the same kind of signaling that changes an entire organism at puberty from a child into an adult. Okay, so 3 different mechanisms by which cells communicate with each other: autocrine, paracrine, and then endocrine. Okay, so here are the slides, Max, that I was alluding to. This one and then the next one. And what I want to point out on these slides is I want to point out sort of this relationship between the cells and the matrix. That's what I'm really most interested in you guys all understanding. And so these circles are high magnification views. So they're high magnification views of like this point right here. High magnification view of this point right here. So let's just orient you. This is the epithelium. That's like your skin. Here's a basement membrane. Here's fibroblasts, these sort of spindle-looking cells, kind of fat in the middle, and then they kind of thin out on the edges. That's a fibroblast. And here's a blood vessel. And so we looked at, with recruitment of leukocytes, we looked at things leaving the blood vessel and migrating out to the extracellular matrix, the ECM. The extracellular matrix, that's outside of the cell, but it's the matrix or the jungle gym that the cell's attached to. It's the substrate they attach to. So let's look to see what is that made of. So first and foremost, if you blow up this basement membrane right here in higher magnification view, you can see a couple of different key basement membrane proteins. You've got these laminin molecules, which look like kind of this cross. You've got type 4 collagen, which is a strand, but it's kind of a helical strand. Then we have proteoglycans. Now, proteoglycans are interesting molecules. Hopefully you learned about these in 201. So proteoglycans are a protein that has a sugar. And if you look into the sugar portion of the proteoglycan, it has a component that has a high affinity for salt. Okay? And salts will bind. And when salts bind, what does water like to do wherever salt is? Water likes to follow. And so proteoglycans are responsible for the hydration capability of tissue. That's why your tissue has fluid, is because of the proteoglycans. It doesn't all get reabsorbed back into the venous circulation. Okay, so this is the basement membrane that makes up this architecture. Type 4 collagen, laminin, and proteoglycans. Now, let's look at this presumable fibroblast. It looks like it's just hovering in space. And I told you, and I promised you, it doesn't just hang there. It's got to be attached to something. So if you look more closely, and that's what this blow-up is, here's your fibroblast right here. And these structures that look kind of like prongs are what we call integrins. And they're molecules that integrate a cell to its outside environment. Okay, there's an alpha subunit and a beta subunit. These integrins physically attach the fibroblasts to the matrix. And what's in the matrix? Well, you still see the proteoglycans. We also see these helices of collagen. There are some other proteins that are found in extracellular matrix, like elastin. So that's shown up here on the board as well. So you've got collagen and elastin, which are the two main structural proteins in extracellular matrix. Now, there's also fibronectin. There's also vitronectin. There's many others that are at like less than 1%. But collagen and elastin make up most, like 95%, or probably more like 98% of the extracellular matrix is made up of collagen and elastin, the two structural proteins. Collagen gives its tensile strength, and elastin gives its stretchiness, its elasticity. So that's why when you take your skin and you pull on it and you let it go, it snaps back. That's because of elastin. And the reason when you pull on it, it doesn't tear off, that's because of collagen. Now, there are disorders. There are skin disorders, okay? One was characterized in a couple of different movies, like in Hollywood. There are disorders where people have a collagen disorder, and instead of having enough collagen, that motion right there would actually slough off the skin. The skin would come with it, okay? So, take-home message from these slides, the cell doesn't just hover, it's attached physically. Now, when it's attached, what is it doing? Well, this is the one I think that might just kind of blow your mind. And you've probably seen something like this in the past. Here's the cell, this is the fibroblast, here's the nucleus. The nucleus is the center that tells the cell, okay, it's time to proliferate, or let's do some protein synthesis. Let's change into something else, or what we call differentiation. Let's go from one cell type to another cell type. Let's move. It's time for us to move. We've gotten signals to move. This is all what the nucleus dictates. Well, the nucleus is actually attached to the phospholipid bilayer, specifically to what we call these FACs, or focal adhesion complexes, and they're attached by a cytoskeleton made up of actin. Okay? Actin is one of the same proteins that's found in your skeletal muscle that allows for actin and myosin to interact. It's the same actin. But inside the cell, this actin, which you know actin actually has the ability to grow and shrink, right? So this filamentous actin is connected physically to the focal adhesion complexes, which are physically linked to the integrins, which are physically linked to the extracellular matrix. So this is how the cell directly communicates with its outside environment, is through these integrin molecules. But they have a direct connection to the nucleus. So if the nucleus says it's time to migrate, it goes up here and it detaches this integrin molecule. It expresses integrin over here, and you attach over here, and that's how it starts to crawl across its matrix, right? Does that make sense? And this happens in your skin wounds so slowly that you can't see it, but day 2, day 3, 4 days, 7 days, 2 weeks later, you're like, wow, those fibroblasts have been migrating. Looks pretty good, right? You just can't see it. Is this kind of cool? Like this is happening like right now while you're here. Because there's repair happening in your body on a regular basis. Okay, we talked about angiogenesis. And I know a couple of you are a little confused on the pre-quiz. Maybe the readings didn't do as great of a job. So let me make sure that we understand what angiogenesis is. So angiogenesis is a term that is the development of new blood vessels from pre-existing blood vessels. Angio, angio root is blood. And then genesis is beginning. That's what the Latin translates to. So this is a blood beginning or a blood vessel beginning. And so you can appreciate on the far left, we've got a tube. It's a capillary tube. And you can probably appreciate or just imagine inside where those endothelial cells are, for that capillary sprout to be coming out, there has to be a gap. So just like when we want to recruit monocytes out into tissue macrophages, we need the vessel to become permeable. We need the endothelium to separate away to create a place for the monocyte to leave. When those endothelial cells pull away and then you tell the endothelium, replicate, they grow another one and they grow out of the hole. Oh, yeah, it's pretty cool. Okay, so if you've got an endothelial cell here and an endothelial cell here, okay, and you're going to replicate this endothelial cell, it's going to replicate out like this. And this is how you get your sprouting. So that previous slide is relevant to endothelium, not just fibroblasts. So in order to do this, you got to tell this guy, make another one, and then you put it here, and as it starts to migrate out, you want to bet, it's not integrins with endothelium, it's cadherin, but similar concept. And so they stretch out in this direction, and that's how you make this new vascular bed. Okay? That's also happening in that wound over like a 2-week period. Okay? And angiogenesis is stimulated by a lot of these chemical signals that the macrophages put out. So the macrophages come in, they make all these signals. Through paracrine activity, it tells the native endothelium, replicate, sprout, make some new blood vessels, because we need to increase perfusion of blood into this damaged tissue so I can bring in more oxygen, not just so that I can make ATP, but I need a new capillary network so that all of the waste products from metabolism, as I'm healing the tissue, I need to remove them. So I have to remove them through the venule side as well. So you don't just make an arterial network, you make a mature network that has the venule side so you can get rid of all the waste products that are a result of an increase in metabolism because you're trying to heal. Okay? I saw a hand somewhere. Okay.
Yeah, so I read somewhere that the reason people that run are healthier is because they have less capillary bed, and that puts less pressure on the heart. But say with someone that's very, say, overweight, they have more of these capillary networks, capillary cells, and that over time accumulates and puts pressure on like the heart. My question is, after this wound healing is completed, you said we have this mature network, right? Now is that permanent, or when that means
It regresses. So let me address the question that's relevant to what we're talking about here, which is a good one. Does this thing stay around? And the answer is once the healing is done, the new vascular supply goes through what's called angioregression. It shrinks away. Okay, we'll see that a little bit later as well. Okay, so let's talk about first versus second intention healing. This is first intention healing, which is this drawing right here. Okay, I don't remember, was it right here up front? It was well characterized by one of your colleagues. Usually you've got minimal scarring and minimal contraction that's necessary because you've got a very thin wound. So you've got a thin wound, You bring in neutrophils. You create a clot, a platelet plug. Now look at what happens to the end of the vasculature underneath in the first week. You get a angiogenic response, and now you've got macrophages here instead of neutrophils. You have a plethora of new vessels that have formed. That's what gives rise to what we call this granulation tissue. Granulation tissue is characterized by a large new vascular supply, and I'll show you histology of it. And then after a couple of weeks, you get rid of the macrophages because they're no longer needed. You have a little bit of a fibrous scar or a union where you brought the two edges or you approximated the tissue. That's what that word means. And then you can see that the vascular supply, to Max's question, regresses. It goes back to normal. You don't need more vessels sticking around for a long period of time. Healing by first intention. Okay? Now, in contrast, let's take a look at second intention healing. Second intention healing, you have a much bigger hole. Kind of, we drew it looking en face or kind of from top-down view. And you're going to have similar types of activities where we've got neutrophils, first responders. Then we have macrophages. You get a much more aggressive vascular response because there's more tissue to heal. So you've got more fibroblasts that have to migrate in, make more collagen. You get a larger fibrotic scar, and you have more aggressive wound contraction because you have a bigger gap that you have to pull together. Now, just a tidbit, scar revision. So there are ways to try to take something like this and convert it to healing by first intention. And so what plastic surgeons will attempt to do, let's say you've got a defect that looks something like this, okay? So oftentimes what plastic surgeons will do is they'll take a wound of second intention shape, they'll come in here with a scalpel, and they remove that tissue, and they bring these edges together to make it look like this. But they'll do that after it heals, oftentimes. So they might let it heal, and then they can, instead of being this aggressive, after this thing heals, right? I should just use the eraser. So this is brought together, it's a little closer, and this looks pretty bad. Okay? But the body has brought this together naturally. So now they'll come in, do their surgical incisions, and bring those two edges together to make it look more like healing by first intention. So that's scar revision. I mean, it's not like I just gave you 4 years of medical school and then 4 years of a surgical residency in plastic surgery, but that's essentially what happens, okay? Make sense? Like why that would work? They'll do it a couple of times. Anytime, the challenge is anytime you have surgery, there's a risk. And so, surgeons are always a little leery of, repetitive surgeries on patients. But, yeah, they would probably revise the scar once or twice, but they're not going to do it like 10 times. They say, you know what, I'm not going to be able to get results that are that much superior than what we have, and it's just not worth the risk to put you under anesthesia and to go through the whole thing again. I know the bottom line is you're going to have some level of a defect here, but it's significantly improved, so I think we just need to go with that. That's kind of what they would probably say. All right, so some of these pics get a little gory. I have a couple of warnings. This one's not too bad, but this is a skin ulcer. And early in my career, I did a lot of work on diabetic wounds. We're still doing work on diabetic wounds, but this skin ulcer is of the foot, and this would be healing by second intention. These often become infected, but many times they'll close. And so we're going to look histologically now. That's the gross image. We're going to look histologically at the wound bed. And so here you can kind of see this large gap between the lesions. You guys can see that? It's pretty obvious. So here's the underlying dermis. Here's epidermis. The stuff looks like it's kind of sloughing off. It comes down here. It comes down here. This is exposed dermis. This is essentially the middle of that. Okay? After about a week, we're assuming this one's going to close. So this isn't this exact foot ulcer. This is an example of what happens in one that closes. This is what we want to see happen. This is histology. This is hematoxylin and eosin stained histo of wound healing in granulation tissue phase. And what I want you to be able to appreciate is just looking at it, it should have sort of an appearance of granular, okay? And what makes it look granular are all these blood vessels. So there's a blood vessel, a circle. Here's a blood vessel, a little bit more of a irregular shape. Here's a blood vessel. Here's a blood vessel. Here's a kind of a long longitudinal slice through a blood vessel. Here's sort of an irregular shaped blood vessel. Do you see all these blood vessels that I'm circling? This is all part of the granulation tissue. And so it's brought in macrophages. The macrophages triggered the endothelium to go through angiogenesis. Now you're flooding this newly healed tissue or remodeling tissue with blood, bringing in oxygen, allowing for a lot of ATP to heal and do everything that you need to do with respect to replicating cells and migrating them into the void space. And then this panel is wound contraction that's taken place, and you've got reepithelialization, so you can see the stratum corneum, which is the top layer, has covered over. Do you see kind of the regions of heavy pink? You guys see all the regions of heavy pink? That's collagen. The pink stains the collagen. So you have more deposition of collagen in this histology, which is where the new tissue has been laid down, and that's the fibrotic scar. So, in some cases with patients, we have an excessive amount of collagen deposition as a result of this wound healing that we just walked through. So if you're kind of prone to keloid formation, what we can say about your skin is that you have a very robust capacity for laying down collagen. So your genes have coded to say, if I get a scar, I want to make sure it closes. I'm not messing around. And so as a defense mechanism, keloid formation isn't necessarily a bad thing. It means that you're quickly trying to close the wound. It does end up leading to an overabundance of collagen deposition, and that's what is the raised portion of the scarring. And so this is a pretty aggressive keloid. Histologically, what does that look like? This should be of no surprise. And the arrows are pointing to more pink collagen being deposited. And you can kind of appreciate the surface architecture where this looks a little bit more bumpy and raised, like what you would expect with a keloid, in comparison to something like this, which is a little bit smoother in profile, except for right here, where that's probably where you're going to see and feel the scar. Okay? So that's keloids. Questions? Not necessarily. So the question is, in patients that keloid, is their vascularization more aggressive? Not necessarily. What's more aggressive is the amount of collagen that gets laid down. Yeah, the fibroblasts are basically like, on overdrive. Yeah. I don't know. That's a good question. I don't know. I wouldn't have any reason to believe that there's more in number, but they're probably putting out more collagen. But I mean, I'm just guessing. I actually don't know. Okay. So at this point in the lecture, I'm going to shift gears slightly because I want to introduce a topic that I've spent a lot of time on. It's pretty popular in literature these days, and it's looking at how stem cells can be used to augment healing of tissues. Okay, and specifically for purposes for today, we're going to talk about skin because that's where we've been. But we're looking now as a field at using stem cells in all sorts of different spaces. And so these stem cells can become fibroblasts. And so you could see that maybe they would be useful. And if you put stem cells into liver and you turn them into liver fibroblasts or hepatocytes, that could be useful. If you put stem cells into skin and they become dermal fibroblasts-like, that might be useful. So there are numerous types of stem cells, and I alluded to this in our first lecture. And so this table is kind of talking more definitively and more detailed about the space of stem cell biology as it relates to where researchers are right now and how it's translating into clinical opportunities. And one of the reasons that I want to share this with you is I fully believe in your lifetime that as healthcare professionals, you'll be making choices or be able to make choices about some of these types of therapies for your own patients. You're starting to see it today. It's still very controversial, but you're starting to see it today. So in the category of stem cells, we have really two main types. We've got what we call embryonic stem cells or adult stem cells, okay? Just two main types. And so what's the difference? Embryonic stem cells come from embryos. And in order to have a source of embryonic stem cells, you really have, it's destructive to the embryo, okay? So you can appreciate over here on embryonic stem cells, whether it's in vitro fertilization or nuclear transfer, the ethical concerns at the bottom of the list is you get destruction of the blastocyst. And so this has been highly debated within the public domain on should we be playing in this particular space, okay? These are the ethical concerns. The reason that we, as a field, are interested in them is because these embryonic stem cells are what we call totipotent. T-O-T-I-P-O-T-E-N-T. Totipotent. That's a word that means these cells can produce all cell types. So you can take an embryonic stem cell, given the right signals, you can make it become just about anything. And so it shouldn't be a surprise. I mean, you take an embryo, it's a single cell as an egg, and then it's fertilized by another cell, which is the sperm, and then cellular divisions happen, and you go from a single cell to two cells, then quickly you go from 2 to 4 to 6 to, and then you get an entire organism. So that power is found within embryonic stem cells. There's no question about that. They're easy to identify and to get, or to isolate is what it says here. And these can be genetically matched to the patient because they're young enough that the immune system hasn't been programmed. So if you're using embryonic stem cells therapeutically, you don't worry about cellular rejection, which is what we're going to talk about next week, okay? Limitations. They're not available at Circle K. It's hard to get them, okay? It's even more difficult now to put your hands on embryonic stem cells because of these ethical concerns, and a lot of governments have restricted access to embryonic stem cells. Another limitation. Often, teratomas are created. What's a teratoma? Teratoma is when you got a mass of tissue that's unorganized, but it's differentiated into a number of different things. So it's a mass of tissue that came from an embryonic stem cell, and it has hair and nails and teeth and bone and skin, okay? But it's unorganized. And I keep hopping back and forth between these two columns. These are both under the category of embryonic stem cells. This one is through in vitro fertilization, so in vitro fertilization clinics where most of the extra embryos are frozen. And so when couples are going through fertilization strategies, they may get 6 to 8 embryos that are successfully fertilized. And they freeze them, and then they decide, okay, we'll do an implantation into the mom, and we'll implant 3, because we don't know if it's common to do multiple implantations because you increase your chance of success. So you do an implantation, there's no guarantee that it's going to take. So you implant 3, you hope you get 1. You implant 3, sometimes you get 2, and then they have twins. You implant 3, sometimes you get 3, okay? You implant 6, sometimes you get 6, and then you get your picture on the cover of a magazine at the checkout stand, okay? So leftover fertilized eggs that aren't being used is a big source for in vitro fertilization, because these are families that said, okay, we had twins, we just wanted 1, we don't want any more, but we'll donate this to science. So that's where they come from, okay? Nuclear transfer, this is where you actually take the egg, okay? You get a donor egg from a woman, and then you transfer a nucleus into it to tell it what to become. But these are all embryonic sources, because these eggs have to come from a donating mom. What's that? You can't make them in the lab. So the biggest challenge here on these 2 categories, ethical concerns and the creation of what we call teratomas, which are masses of tissue that won't amount to anything. And so I would offer, in the field of stem cell research, this area has gone out of style. It's gone out of vogue. People aren't playing in this space. I've never spent any of my career in embryonic stem cell research, but now fewer and fewer people are in this space, because I think what we're discovering is there potentially is too much technology within the embryo that mankind doesn't really know how to navigate it, and it doesn't turn out very well. Okay, over here, adult stem cells, and this is where everybody has moved to. And this is where I've spent all my time in stem cell research, is on the adult side. Not controversial, because all of us have adult stem cells. Adult just means on the other side of being born, okay? You don't compromise the embryo. So cord blood stem cells, placental-derived stem cells, bone marrow-derived stem cells, adipose-derived stem cells, all of those are adult stem cells. So you take the cord after the baby's born, and you strip out mesenchymal stem cells from the umbilical cord. Well, the baby is fine. It's already born. So no ethical concern, and most oftentimes people will discard that tissue as a biohazard, because there's nothing, you can't do anything with it. Same with the placenta, or what we call the afterbirth, when you get stem cells out of placental tissue. The younger the adult tissue, like cord and placenta, the more pluripotent, not totipotent, but pluripotent. Pluripotent means that it has the potential to become many, many things, but maybe not everything. So you're down a little bit in the pathway of differentiation, where you can't make an entire organism, but fewer things can go wrong too. So in stem cell research with adult-derived stem cells, even the youngest stem cells that are placental or umbilical cord, we don't see teratomas forming. Yeah. So the field has kind of migrated to be right here. Less controversial, much more abundant. Okay, we can get it out of patients that are at any age, because you can pull it out of blood, you can pull it out of bone marrow, you can pull it out of fat. Okay. So, embryonic stem cells, and there's a side category that I want to introduce on this slide. This is all the same information of how embryonic stem cells are created, and then here's our adult ones down here. Then this category of induced pluripotent stem cells, they were popular, and then they've kind of like stalled out a little bit. They were kind of this halfway in between embryonic and adult, and they're induced pluripotent, meaning you take an adult stem cell, and then you use transcription factors, and you inoculate it with transcription factors that make it behave more like an embryonic stem cell. So you don't have to compromise an embryo. You can take a young, let's say, umbilical cord mesenchymal stem cell, you inoculate it with transcription factors that are embryonic-like, and then that cell starts behaving pluripotent, more like an embryonic stem cell. Okay? maybe not totally totipotent. So it's kind of like a half step between pluripotent and totipotent. Sorry, Max, go ahead. Here, they haven't reported the same probability of teratomas as embryonic, so these were a lot more popular, but they're a little bit more difficult to generate because you have to add these genes and push them down a lineage, and sometimes they behave and sometimes they don't. So most researchers now kind of, just moved on to young adult sources. Like umbilical cord blood and placental tissue is really popular right now. Right? I mean, everybody's doing it. Okay. So what I want to share with you in the remaining time is one little study that we did. This was a grad student in the lab where these were adult-derived stem cells, and we pulled these from lipoaspirate. So liposuction, you take the liposuction and you wash it, and then you digest it with a collagenase, and you separate the different columns, and you get what's called the stromal vascular fraction. In this SVF, or the stromal vascular fraction, contains these adipose stem cells, the ASCs. These ASCs have the ability to differentiate into fat, obviously. But they can become bone, muscle, cartilage, skin. They can become a lot of different things. Remember, they're pluripotent, but maybe not totipotent like with an embryo. So we take that lipoaspirate, we get our stem cell population, and we have to make a scaffold. Because I told you guys, so half of my career has been spent looking at cell biology, and then the other half's been looking at matrices or architecture, because the cells have to live in an environment. And so there's two big knobs that we can turn. What we do in my lab is a lot of translational medicine type of research. So we're trying to build stuff, and I want to put it into people and help cure certain things. So this is making a matrix, but we use a process known as electrospinning. This electrospinner is a high voltage power supply that the positive charge is connected to a needle. The needle is connected to a tube that's got a slurry of protein, okay, like a solution. It would be like a protein shake in a way, okay? So you take extracellular matrix proteins, put them in solution, stick them in the syringe, you inject it at a certain flow rate, it goes to a positive charge. This is the negative charge target. This is 40,000 volts. Yeah, this is kind of fun to watch. I'll show you a video. And there's a gap here between the electrodes. And so you charge this protein, and then it flies across and hits this target, and it makes a scaffold. Here's what the structure looks like, and here's the little video sequence. So it kind of comes out looking like you're spinning spider webs. And this is called electrospinning. This has been in the field for a long time, but we can actually make architecture that these cells can live in that look like the matrix of skin. And in so doing, we can build a replacement scaffold or architecture. This one here was captured with a high flow rate, and this one was with a lower flow rate. So you can see open versus closed architecture. And these spaces, the black spaces in between what look like the spaghetti strings, this is where the cells will live. The cells will squeeze through those spaces. And over the next few days, I'll show you pictures of these kinds of structures for you to be able to appreciate what this looks like. But here's a scanning electron micrograph after we basically tried to make a skin patch in the lab using these human adipose stem cells. So you've got the bare scaffold in the background, and this is an adipose-derived stem cell that's sitting underneath a blanket of matrix that it's made. So they make their own matrix, these stem cells. And it's coating. You can see half of this is coated up here, and the other half here is not coated. It's bare. And then here's an adipose-derived stem cell that's actually dividing. So it's bunched up, and it's about to become two. So we're pretty happy with this. So what we decided to do, these were human adipose stem cells. And so we went into a SCID mouse, and you're going to learn what SCID is next week. This is a severe combined immunodeficient mouse. So it doesn't have T cells. And by lacking T cells and B cells, there's no immune system. So I can take a human cell and implant it into the mouse, and the mouse won't reject it because it doesn't have a functioning immune system. Okay? These are like bubble mice. You have to have them in special cages. But we were making something that we wanted to evaluate the humanness side, and so you utilize an organism that allows for that. So the study endpoint was 6 days. We created these wounds on the back of these animals. Okay, this all has to go through approval through a committee in order for you to be able to get these experiments done. And we took these scaffolds that had the stem cells that were growing, and we grew them for a few days so that you could see the matrix on the scaffold. And then we implanted those. And here's our control group. Here's a scaffold made up of elastin. Tropoelastin is just the precursor to elastin, but you can consider this elastin. And then this one's collagen. So we've got the two main structural proteins plus the stem cells compared to control. Now, control was standard of care therapy. It wasn't just like we left the wounds open, and put our heel in it or something. We kept them clean. We used antibiotic ointment. But what you can see after 6 days, these wounds are still open. 63 to 75% still open. Healed, but not completely closed. These scaffolds with the stem cells were almost, in some cases, they were completely healed after 6 days. So these control wounds go on, and it takes them about 12 days to totally close, just for reference point. So we're able to cut healing time essentially in half by delivering a scaffold and a cell type that's a stem cell, okay? Why stem cells? Well, it's harder to find fibroblasts. It's harder to collect fibroblasts. You collect the stem cells, they can become the fibroblasts. So the last few graphs are just kind of characterizing the results. The wound closure was significantly faster in the scaffold group compared to the control group. If we look at the thickness of the epithelium to give us an indication of how well it healed, we saw statistically thicker epithelial scores in our scaffolds plus our cells. And we published this work, and this student went on to medical school. The picture of that guy in the surgical glasses was a former graduate student who went on to medical school. So Wednesday, this is where we're going to pick it up. And I want you guys to look ahead. There are some gory pictures coming, but I want you guys to look ahead to see what's coming so you're not shocked. They'll look less scary on the big screen if you have a chance to preview them. But take a look at the major phases of wound healing, and we're going to talk about the ways that we move from one phase to another on Wednesday. Okay, and then I'll finish up with a regenerative medicine lecture at the end of Wednesday. Thanks, you guys. Have a good night.